So today, Lab 12, one exercise, bacterial transformation using the PGLO system. So this is a kit that we buy from Biorad, which is a biotech company. So he may ask, why transform, right? Why would we want to change things? Well, for better understanding of what we deal with. So we're going to be dealing with transforming bacteria, but they've done this all the way up to eukaryotic organisms like these mice. You'll notice that 2 of them are glowing green, right? You see how their eyes are glowing green, even their tails. That's because they've been genetically altered to produce this green fluorescent protein. So under UV light, you can see it. But they're not doing it just to make glowing animals, right? They're making it because it's kind of a tag to let them know that the genetic change that they tried to institute worked. So in addition to the green fluorescent protein, they gave them some other ability, right? And you can't see our DNA, right? You can't look at this mouse and see that one's DNA is different from the other. But you can see if you genetically give them ability they don't normally have, like to make green fluorescent protein, you can very easily see and visualize that they do have that genetic information because they are making the green fluorescent protein. Unlike this guy, he didn't genetically change, right? Something went wrong, and he's not glowing green. So they wouldn't be able to use it for their research because it doesn't have the genetic information they wanted it to have to be able to study it. So a little bit easier example to understand that's really cool. And I would even show this to my kids that I taught over the summer. Whenever you say the word sperm, everyone pays attention, right? So this is actually a video. It's short, but it's pretty cool. Hopefully it'll go to that website for me, yeah. So what you're seeing here are sperm in the reproductive tract of a female fruit fly. And female fruit flies will mate with more than one male fruit fly. And this is so that hopefully her children are genetically diverse, right, and more likely to succeed in the environment. So they have found that the sperm themselves from the different males compete, right, because they want to get to the egg. And then they did further testing and found that the female reproductive tract actually produces chemicals that tries to stop this competition. But the reason why we're able to visualize this competition, you can really see the green ones, right? But look closely. Do you see the red ones? So one of the male fruit flies, they altered their sperm to produce the green fluorescent protein, and the other one they altered so they would produce the red fluorescent protein. So you could tell which sperm came from which fruit fly. And you can actually see, right, as I said, there's competition happening between the sperm. Right? So pretty cool. So in this case, it's helping us, be able to visualize, right? These green fluorescent proteins can be very helpful in visualization of what's going on. And there's a difference right down to the genetic level, right? These 2 sperm are genetically different from 2 different fathers. So I used to have an article pinned up in here, but it walked away. They've used this type of research a lot. There was a really cool article that was out a while ago in Science Magazine about how they were using green fluorescent protein to help study feline leukemia. It's been used for a lot of stuff because, again, it helps us visualize different organisms. Then you can also have fun with it, right? So this literally is bacteria growing on an agar plate, and it's different bacteria that have different fluorescents been added to them, different fluorescent proteins. So there isn't just green and red. There's a whole bunch of different colors out there. Some of the kids this summer literally drew with bacteria. This isn't one of their products. This is something I found off the internet that someone else had done. But they did some cool stuff. So this is actually literally bacteria growing on a petri dish that they used to make this drawing. So what is this process of transformation? This is taking genetic information from one organism and putting it into another. And this happens a lot among bacteria, right? Where genetic information from one can be picked up by another. Typically, the cell that donated it died. Today, we're not going to start with 2 cells, instead just a single bacteria, and we're just going to add DNA all by itself into the test tube. But that's still transformation. They're still picking up DNA and bringing it into their cell, and if they utilize it, they can change. So in my lecture class, I use the analogy of transformers, right? We all know what transformers are, right? Go from a car to a robot. My son's convinced that someone has a Camaro that looks just like Bumblebee that drives around New Orleans. My son is convinced it's going to turn into a robot. He screams Bumblebee. Every time he sees it, I'm like, it's not going to turn into a robot. It's just a car. But no, it's not Bumblebee, right? But who knows? Someday, right, we might really have transformers, right? Who knows? But we can do this with organisms, right? We can transform them, right? Think about that change, that transformation, right, is truly what we're talking about. And when it comes to organisms, that information that determines what we look like and what we can do is in our DNA, right? That's the information. So if you give that information, if you give that DNA to an organism, depending on what that information is, they can use it and change according to that information. So for today, the information, one of the bits of information we're going to give them is how to make green fluorescent protein. Right? We're going to give them the DNA, the information, and they're going to make green fluorescent protein. So under UV light, they're going to fluoresce green, right? After we transform them and grow them. So transformed cells take up a portion of the DNA from another organism and integrate it into their chromosome. The type of transformation we're going to do today, it's a circular piece of DNA known as a plasmid. And they're just going to take it into their cytoplasm and utilize it. They're not going to incorporate it into their genome. But sometimes this will happen all the way to this level where it literally incorporates into their chromosome. So this is an active area of research because I said this happens naturally. But we've been able to figure out how it happens and cause it to happen in lab situations. But it has happened over time. And unfortunately, it's led to a lot of resistance to antibiotics because how the organisms are resistant is dependent on their genetics. What can they do, right? What information do they have? What abilities do they have? So Klebsiella pneumoniae is resistant to a class of antibiotics, and the reasoning behind that, right, it can destroy this penicillin-like drug because it has gained a plasmid, a circular piece of DNA, that contains a gene known as BLA, and in this case, this additional marker to that gene, which stands for New Delhi metallobeta-lactamase. The BLA stands for beta-lactamase. This is an enzyme that breaks the beta ring that's found in ampicillin, penicillin, those types of drugs. Right? And so today we're actually going to make a superbug too. We're going to give it BLA. We're going to give it beta-lactamase enzyme so that it can destroy penicillin-like drugs. The drug that we're going to use is ampicillin. Right? So we're going to make it so that they can grow in the presence of ampicillin, the ones that we transform, the ones that we give the DNA to. But this is happening in nature, and as you can imagine, those organisms outside of this lab create serious problems for us because we can't treat with these antibiotics if they have these enzymes that destroy the antibiotics. So this movement from one bacteria to another is referred to as horizontal transfer. And this can even happen between different species under certain circumstances. So one bacteria to another. And there are several examples where we don't know exactly how it moved. Was it a cell died and the other one picked it up by transformation? Did the cells actually join by conjugation, which is another horizontal gene transfer? Or was it transduction? Did a virus pick up the DNA from one bacteria and transmit it to another? We don't know exactly how it got there, but somehow it got from one bacteria to another, this genetic information. And so that's horizontal gene transfer. Vertical is where it goes from mom to children. So again, once an organism gets it, especially if it's advantageous, it will pass it on to all its progeny, and that's vertical transmission. So that's the way in which genetic information moves throughout a population. So in 2010, they had isolated 180 isolates, different bacteria that contain beta-lactamase enzymes, right? This gene specifically to be able to destroy penicillin-like drugs. But of course they remained susceptible to other classes of drugs that work in different ways, right? So several other of the drugs are still effective. We've talked already about MRSA, right? Amygdalin-resistant Staphylococcus aureus. And again, it's because of genetic information that they can destroy ampicillin, penicillin-like drugs, and therefore we can't use that against them. We have to use other types of drugs. Tuberculosis we have strains of it that are extremely drug resistant. So most of the antibiotics that we use against tuberculosis, it's resistant to. Right? So we're limited in what drugs are effective at killing those strains because they've developed genetic ability to be resistant. E. coli 0157 is probably one of my favorite examples. That particular strain of E. coli has the Shiga toxin. The Shiga toxin is what makes Shigella very pathogenic. It's a toxin that literally deteriorates your intestines. It kills the cells lining your intestines. So E. coli doesn't normally have that, but there is one strain out there identified as 0157 that has the Shiga toxin. It got it somehow, probably from Shigella. Some type of horizontal transfer, whether it be conjugation, transformation, or transduction. But it has it. Right? And that's where it came from. Right? Shigella was the first to have it. Yeah. So Shigella is similar to Salmonella. Most people are familiar with Salmonella. Shigella and Salmonella are, two really bad gram negatives and cause pretty serious food poisoning. So in order for transformation to happen, the cells have to be competent. Only about 1% of the known bacteria are normally competent, that are receptive to picking up DNA from the environment. So transformation in nature is very complex, right? It's a developmental process. Expression of about 40 genes is required for competency, for them to be able to be transformed. And it requires energy to take in this DNA. Right? They have to expend energy to actually do this. But under certain circumstances, this could be very beneficial for them, right? Especially if you're E. coli, right? And you pick up that Shiga toxin from the environment, you just became really powerful. It's evolved in a way for DNA repair because bacteria are prokaryotes. They have a single set of most of their genes. So if something happens to their DNA and they can't repair it, if they really need it, they'll die. So this is one of the ways that they could repair their DNA if they picked up DNA from the environment. They could potentially repair themselves. And this happens when they're under stressful conditions, when there's a lot of cells, right, or there's nutrient limitation. It can be viewed as a primitive means of sexual reproduction because all sexual reproduction really is exchange of genetic information. Bacteria don't do this when they reproduce, right? They reproduce by binary fission. They basically just have moms. They're copies of their moms, right? But when you're exchanging genetic information, either by transformation, like we're talking about here today, or conjugation, that one's sometimes definitely referred to as bacterial sex because the two cells literally join up and exchange genetic information. What information only goes one way, though. Right? But that's the same for humans. Sexual reproduction is the exchange of genetic information. All sperm are is mobile DNA, right? That contributes to the forming of a new organism. So we can artificially make cells competent, right? Make them receptive to picking up DNA from the environment. You can do that through electric fields and makes the membranes permeable. Cells can take up chemicals, drugs, or even DNA in this state. So this technique can be applied to cells including eukaryotes. So not just bacteria, prokaryotes, we can do this to eukaryotes as well. It's very costly though. You need special equipment, right? Special vessels for holding the organism. So we don't do that here at Delgado. I don't know, in the biotech they might. Who knows? But we use a little bit simpler system. We use chemically treated cells. So we have a special line of E. coli that we utilize in the lab, and we use a special transformation solution of calcium chloride. So the little tubes that I'm going to give you guys today contain the transformation solution that contain calcium chloride. And this helps with the permeability of the membranes to help get the DNA into the cells. It's thought that that the positive calcium ions bind to the negatively charged groups of the phosphates of the membranes, right? Thus shielding the cell surface from the negatively charged exogenous DNA. So remember, DNA is negatively charged. So this helps it get into the cell instead of being stuck to the outside of the cell. So only one strand of the DNA is allowed into the host cell, while the other strand is degraded outside. However, they'll repair it. Once it gets inside, they're going to remake that other strand. And that's too when they're trying to find a place to put it in the chromosome. They'll find a place, they'll actually bump stuff out they already have there, and then they're going to make it double-stranded. So it enters in single-stranded, finds a place to go. In the case of what we're doing today, it's going to be a circular piece of DNA that stays by itself. So again, there are kits that you can buy from different industries. This is done a lot. When I was doing molecular research in the microbiology lab at Southeastern for my master's degree, I utilized these types of cells. And one of the main reasons why I was utilizing it was a cheap, easy way for it to copy DNA. Right? So I had segments of DNA I'd gotten from frogs, and I needed to replicate that in order to then sequence it. And the way I did that was give it to the E. coli and have them copy it. Then I'd lyse the cells and get the plasmid back out, and then utilize that to sequence the DNA. So there's different levels of efficiency, how well, right, how often will they actually accept it. So not every single cell in our solution is going to take it up, but because we're using specialized cells and all the mathematical calculations have already been figured out for us, we're going to have a really high efficiency, which means a lot of the cells are going to get transformed. So we are going to, at one point, to feed our cells, right? So we're going to give them food. And in case, look at this one, of course, glucose, as we know, right? Really good food source. So we have a special type of media that will feed the bacteria, again, to put them in the best possible conditions for this to work. And for this one, they have a positive control. We'll have controls in our test as well. So for this system, this bacterial transformation, what it is a process of bacterial cells, right, taking exogenous from the environment DNA that's not normally part of them and actually taking it inside. Only competent cells will do this. These are ones that have the ability to take up the DNA. One of the things that we're going to generate is bioluminescence. Bioluminescence is when light is emitted by an organism. Some of them can make light on their own. Others will absorb energy and light energy and bounce it back. So like you could say re-reflect it. And that's the case that happens for us. And these genes, these fluorescent genes, actually come from jellyfish. A lot of jellyfish are fluorescent. So under UV light, they fluoresce. And so if you ever go to the Aquarium of the Americas here, right, down in the section where they have the jellyfish, you'll notice that their tanks are kind of dark. And part of that is so that you can see And I think a couple of them, they do actually have fluorescent light so that you can see. There's one of them that's one of my favorites that's not fluorescing, but it's really pretty. And if you actually read the placard next to it, they have tiny little cilia-like projections on them. And when they move and beat them, it refracts light just like a prism does. And so it looks like it has a rainbow running along its edge, and that's just refraction of light. That's not bioluminescence or fluorescence in that case. But there are lots of fluorescent type organisms out there, especially in the oceans. So this is what happens is that when the light, the UV light is shined on it, and we can't see UV light, right? But they absorb it, their electrons get moved, and then when the electrons move back, they emit the light. And because of the particular protein and how it's absorbing and re-emitting the light, in this case, we see green, right? And it goes within the visible spectrum of light that we can see. So, Peter's a big fan of math, right? So, as I said, all math, it's hard for me to explain that to you guys, but I will try if you're interested in how you could actually calculate this out. But the important thing to remember, as I said, we can only see light within the visible light spectrum. UV light is not within our spectrum, right? And in this case, they're absorbing the energy from that, and they're bouncing it back in the form of light, in this case, in a green light spectrum, which is less energy than UV light, but again, we see it. All the conditions have been worked out, as I said, for today, right? There's no worrying about this. This is a kit, right? Just about every time we do it, it works. So we know how many cells to use. We know how much DNA to use. We know the transformation efficiency. About how many transformed cells we'll get per milligram of DNA we add into this solution. So that means that we know the volume of the mix. So again, in those tubes, I already have a set volume. We're going to spread a particular amount, just like as we did before. So we could actually calculate it like you guys did if we actually spread them out good when we did standard plate counting, right? When we did the dilutions and all of that, right? Because we know all the volumes that we're dealing with for today's experiment. And doing that, we should get about 75 transformants per each one of our plates, right? But we're going to be using little plates, so it's not necessarily easy to count. Normally, you'd make serial dilutions and plate a variety of concentrations to get an accurate count. Again, we're not actually going to count these, but we could, right? Because we know all the parameters. We are working with set volumes. So, for gene expression, as I said, we're giving them a specially designed plasmid called the PGLO plasmid. On this plasmid, right, this is DNA, is genetic information. So there are different segments that give it different abilities. So the beta-lactamase, right, abbreviated BLA, is going to allow them to produce an enzyme, beta-lactamase, that's going to destroy the ampicillin that we have in our plates. So if they transform, if they take up this DNA and they make that enzyme, that protein, they'll destroy ampicillin. The other thing that we have is the green fluorescent protein, as I said, from jellyfish. And that's abbreviated GFP. So that genetic information is right here, right? When they copy that, they're going to make that protein. When you shine UV light on them, they're going to fluoresce green. But this type of gene is under what we call gene expression regulation. In order for that to happen, we have ARAC, which is a regulatory protein. So they have to make this protein. So when they make this protein, it actually allows the cells in the presence of arabinose to actually copy this information. When it's not present, they can't copy the information. And so constituently expressed means it's always made. So both BLA and ARAC, it's going to make it right when we give them the DNA and we feed them, it's going to start making those two proteins right away. right? It's not going to express GFP yet, or maybe not at all. So operons are a sequence of genetic material. They consist of an operator, a promoter, and 2 or more structural genes. Structural genes means it makes something, like a protein, that can do something, or the enzyme in this case. The operator is a segment of the DNA which the regulator binds. So some molecule called the regulator is going to bind to the DNA at that site that we call the operator. Inducers, right? Operators that induce cause something to happen, right? Induce, they cause it to happen. So in the case of the arabinose operon, which is the operon we're using, the protein derived from ARAC gene, that protein that's made, is going to bind. Then arabinose is going to bind to that. And that turns on this situation. So that now the promoter site, which is the region where RNA polymerase binds, will be able to bind. So RNA polymerase normally cannot bind to the site it needs to, this promoter site, to make green fluorescent protein because of the operator. So RNA polymerase, as the name implies, right, makes RNA from DNA. And you've got to have messenger RNA in order to make proteins, right, in order for the ribosomes to make the proteins. The structural genes are other things that they will make. In this case, it's green fluorescent protein. And that's the one that's going to allow us to fluoresce. So arabinose is a 5-carbon sugar. It's found in plants. It's a pectin part of plants. And normally organisms would break down, maybe ferment, maybe use it as an energy source for aerobic respiration sugars. So in order to do that, organisms have structural genes, which would be enzymes that would break down arabinose and even maybe produce ports in the membrane so they could bring more arabinose in. Another operon that's commonly used for E. coli is the lac operon. This is for lactose digestion. Lactose for most E. coli isn't always available. So it isn't always making the enzymes it needs to digest lactose. It only makes those when lactose is present, because it's usually not present. But if it is, they'll utilize it. So in order to do that, they have to have some way of signaling, okay, I need to make these enzymes because lactose is here. So the same thing with arabinose. When arabinose is present, it binds to the operator. So there's your arabinose, the effector, in this case the inducer. It allows the promoter region to be available. RNA polymerase binds, and it would make the enzymes to digest arabinose. But what they've done is genetically engineered. They're using this operon, right, this whole system, but instead of having the enzymes to digest arabinose, they plugged in green fluorescent protein. So when arabinose is present, it binds to the operator, the promoter becomes available, RNA polymerase copies the green fluorescent protein gene into a messenger RNA, the proteins transcribe it into the green fluorescent protein. But only under these conditions, because we actually have control over our DNA, right? Red blood cells make a ton of hemoglobin. That information on how to make that protein is in our DNA. But only red blood cells use that DNA. Your liver cells have that DNA on how to make hemoglobin. They just don't use it because they're not told to. They don't need to. For their job as a liver cell, they're utilizing other genetic information, making other proteins necessary to their job. All right. So within the environment of our body, that's how we end up with different cells, even though genetically they're all the same. They're just not using it all. For organisms, it allows them to adapt to different environments. If they have that genetic information that allows them to digest lactose, when lactose is available, they're going to use it. But they're not going to keep these proteins around when they don't need them. Why would you chock full one of our liver cells with hemoglobin? That would be a waste of energy, right, to have that protein when that's not that cell's job. Does that make sense to you guys? So, in order to have the expression, in order to have green fluorescent protein, you've got to have its expression, right? They've got to take in the DNA and they've got to use it, right? And under these conditions, they're only going to use it if that arabinose sugar is present. So I'm not going to go over the hypothesis statements. I'm going to skip over those. So BLA, as I said, is the enzyme that's going to destroy beta-lactamase drugs. In this case, we're using ampicillin. So we're going to make ampicillin-resistant E. coli today. We're not going to let it leave the lab. So before transformation, do they have this genetic information? No, right? We saw this before, right? We used ampicillin discs against E. coli and it killed the E. coli. So no, but after transformation, again, transformation means we're giving it to them. They are going to have this gene if they're truly transformed. So the first step in our procedure, which is already done for you, you'll have tubes labeled plus and minus. One we're going to add DNA to, one we're not. Why aren't we going to add DNA to it? Control. It's going to be our control. It's going to be our negative control. In each tube, they already contain 250 microliters of transformation solution. This is that calcium chloride solution. I already put them in the ice for you guys and filled it up with water, so they're all chilled and ready to go. Those racks that are in there are floating racks because later we're going to move them to the water bath. You'll see that's turned on next to the refrigerator back there. So they're going to float. Steps 4 and 5, you guys are going to use sterile plastic loops so we don't have to get out the Bunsen burners again. And this bacteria is very sensitive to heat, so we don't want to accidentally have too hot of a loop and kill them because then it won't work. So you're going to pick up a little bit of the DNA and you're going to put it in, a little bit of the bacteria, you're going to put in the DNA positive tube and the negative tube. Because of the way we're doing this, right, they're not going to both be exactly the same. Like we could put them all into the 500 and then divide it into 2, right? That's an extra step. That isn't necessarily necessary for our experiment, but to be consistent, right, you would probably do that. We're not going to. I'm going to come around and show you guys the plasmid solution, which is just the DNA. So when I shine UV light on it, is it going to fluoresce?
Yes.
It's DNA.
Oh, no.
Only the protein fluoresces, right?
Yes.
So it shouldn't fluoresce because it's just DNA. If it fluoresces, it's contaminated with some protein. Okay? And we definitely wouldn't want to use that. So we want to make sure that it's just DNA. It's just the protein that glows. So, of course, I'm going to put 10 microliters in your plus only tube. We'll mix it up. The loops are calibrated to 10 microliters, but it's really hard to get that tiny bit of solution out. So I'm going to use a micropipetter, like I showed you guys before when we were doing dilutions. What should you add if we were going to be consistent? What could we add to the negative tube instead of DNA?
Water.
Water, yeah. We could easily just add some water. Which, again, I'm not going to bother with that extra stuff. It's such a small amount. It doesn't make a huge difference. So, before transformation, we've already said they don't have beta-galactanase enzyme. They don't have the genetic information to make that enzyme. They also don't have the ability to make green fluorescent protein. But after, they're going to have the ability to make the enzyme, and they're going to be able to make green fluorescent protein. So the next step is we're going to leave the tubes on ice for 10 minutes. And again, this is going to give it time to interact with the solution and the DNA, hopefully get into the cells to facilitate this. Then you're going to take your whole rack and carry it to the back. In your protocol for the lab, it tells you to label your plates. Your plates have already been labeled for you. Because each plate contains different stuff, it's easier for me to just label them when I make them. You're going to bring your ice bucket all the way to the back. You'll see that that is set at around 42. It's saying 43 right now, but we'll look at the thermometer. Don't walk away, because you're just going to pick up these racks from the ice and put it right into the water bath, and you're only leaving it in there for 50 seconds. 50 seconds goes by really quick if you walk away, right? So stand right there, time it, pick your rack back up, plunk it back on your ice, carry the whole bucket back to your lab bench. Then once you get back to your lab bench, right, we're going to leave it on that ice for 2 minutes. This is referred to as heat shock. Why would we want to do that? We're going from cold ice water to equivalent to a hot tub when you go on vacation, right? You ever jump from the swimming pool to the hot tub? Yeah, right? That's kind of shocking to the system, isn't it? So it's shocking to these bacteria, so it's a stressful environment, right? And remember, stress is one of those things that's going to help them, encourage them to pick up this DNA, right? It's going to make them more permeable, and hopefully that DNA in that circular plasmid form goes in. And the time really matters, right? Again, you don't want to walk away. They've done studies. It's most efficient if you do it for 50 seconds. If you leave it in there longer, the efficiency significantly drops off. And I've screwed up many experiments in lab trying to do too many things at once and walked away, right? And then I was like, oh, crap. And I had to start all over again. So with this system, 50 seconds. After 2 minutes, you're going to just take the whole rack out of the ice and put it on your lab bench so that now we're at room temperature. We're going to use a sterile plastic pipette. And in this case, we're not going to bother with those crappy pipettes because it's really hard to measure this. I sterilized us some pipette tips, and we're going to use the micropipetter. And you're going to transfer from a little tube that I'm going to give you of LB broth food. You're going to transfer 250 microliters into each one of your tubes. You're going to feed your bacteria. So having done that, we're going to, and again, you don't want to put it actually into the tube, right? So we can use the same tip. So we're not going to use those guys. So we're going to let it sit for 10 minutes. Why are we doing that? Let them eat, right? So what are they going to do with that food we gave them, though? What are they going to make? There's only 2 genes they can make, right? They can't make the green fluorescent protein because the LB doesn't contain any arabinose sugar. So they're going to make the BLA, which is the beta-galactanase enzyme, and they're going to make the regulator, they're going to make ARAC. That's what they're going to make in those 10 minutes. And they really have to make the beta-galactanase because we're going to plate them on plates that contain arabinose. So if they didn't make that enzyme, guess what's going to happen to them when we put them on those ampicillin plates? They're going to die. Right? So we have to give them time to make the enzyme so that we don't kill them when we plate them. And this, of course, is true for our transformed cells. Right? They're the only ones that are going to be able to make it. So then you're going to transfer 10 microliters of each sample onto plates. And again, I'm going to give you guys pipette tips. I'm going to give you little micropipetters that you guys are using again next week. These are preset to 50 microliters. So in order to do 100, how many times do we got to squirt? We got to draw up and squirt onto the plates twice, right? 50 plus 50 is 100. And then you can do both your DNA plates with the same tip, right? And then you'd have to change your tip for your 2 negative plates, right? 2 squirts onto each plate. And then someone else in your group needs to take a sterile loop and spread out on your 2 positive plates. You can use the same loop because they're both the same, right? and then someone else with another loop spread out the bacteria that was put on with a different loop for the two negatives. Don't tape the plates together like it says to do in the protocol. I like to keep them separate so I can spy on them easily the next day. Alright, so what information is going to be provided by the BLA DNA negative plate? So this plate is just food. And it's just bacteria that we started with, right? We didn't add any DNA to it. So this acts as what for our experiment? A control. A control that our organisms are capable to grow on this media, that we started with living organisms. So we should see nice, good growth, and that heat shock doesn't kill them, right? Now, we're going to take the DNA negative, right, so no DNA, and put it on some ampicillin plates. What's going to happen to the bacteria when we do that? These ones that didn't receive the DNA, they're going to die. So this is our control, too, to let you know that ampicillin will kill the E. coli if they don't have the DNA. So this is going to verify, right, that E. coli before we started was not ampicillin resistant. It was only after they were transformed that they were. So transformation could occur only if the PGLO plasmid was introduced into the solution. Which plates are going to show that? Well, it's going to be our positive plates, right? Our DNA positive plates got the DNA. We're going to have two different types of plates. We're going to have one that contains ampicillin and one that contains ampicillin and arabinose. So why does the ampicillin arabinose DNA positive plate fluoresce, why the ampicillin DNA positive plate does not? What's the difference between these two plates? The arabinose. Because remember, you've got to have the sugar, right, that inducer, in order to produce green fluorescent protein. Are the ones that are on the LB ampicillin plates DNA positive, they still transformed? Yeah, because otherwise the ampicillin would kill them, right? So they're still transformed bacteria, but you've got to turn on the green fluorescent one. We have that within the operator system. So it's not, that operon, it's not going to turn on without that sugar. So only one of your plates is going to glow, right? So we have 4 plates already labeled as such, so make sure that you guys put the right solutions on the right plates, right? DNA negative, DNA positive, right? Some of them contain ampicillin, some contain ampicillin and arabinose. So, again, controls for our DNA negatives, for showing that ampicillin does kill and the bacteria can still grow, right? To show just transform cells, but only this one is going to glow because it has the arabinose. And this is hopefully what they'll look like next time. So you'll have the one that glows because it has the arabinose. Your DNA negative plate, which isn't labeled here, won't have any growth on it. And I think it's this one. You can see how there's bacteria here? So these are transformed. This is a complete lawn of growth of bacteria, so you can't see it really well. We did this under the UV light, though, so you could see the glowing green.